what is it that mediates cellular memory? Because a lot of the time you're talking about an exposure, some sort of an environmental exposure that imprints itself upon the cell, and the cell changes its characteristics in the long term. So if you have a cell which is just sitting there, it doesn't divide, it's circulating around the bloodstream, or it's sitting in the brain or in the kidney or something like that, and it has a lifespan of 20 years perhaps, it's very easy for that cell to retain a memory of a prior event because it's not going away anywhere. But other cells cycle all the time. So the way that you would maintain a memory in each case is probably quite different. And we have to divide our thinking, first of all, along those lines. Then the next question is, how is it that we normally respond to a stress, whether it's a long-lived cell or a short-lived cell? And the response to a stress is through regulation of gene expression, these transcriptional regulation, as it's generally called. And the transcriptional regulators starts to get interesting again, because the mediators of these transcriptional responses include the things that we're loosely calling epigenetics, such as DNA methylation, the modifications and organization of the chromatin, and perhaps microRNAs as well. So the big question in epigenetics is not really how genes switch on and off. It's how, once you've undergone that cellular response involving gene regulation changes, how that persists in the long term, how you remember that you switched those genes on or off in the past in such a way that it has changed the cell's ability to switch them on or off again in the future. And that to me feels like a much better depth of epigenetics and a much better way of thinking about it in terms of the response to things like environmental or other toxic exposures. This transgenerational idea is enormously intriguing and comparably unsupported, I think is a fair way of putting it. There are some studies which certainly suggest that there could be transgenerational inheritance, where some sort of mark is not only present in your body, but you put the similar mark in your gametes, and when you form new gametes in the next generation, going through intermediate cells that do not resemble gametes in any way, those gametes, male or female, can then pass on the same mark. It's certainly plausible that mechanistically that might happen. The evidence is weak, and the evidence is probably better in plants than it is in animals. But plants have a lot of different ways of doing this kind of thing that we don't. And one of the ways is through these non-coding RNAs. They have a whole machinery for propagating these things from cell to cell, and presumably from generation to generation. I think where we are with human and mammalian systems is in this kind of murky state of it might be happening, it might not. Some studies support it, some do not. The weight of the evidence is negative right now, but nobody's saying this is not worth studying if you follow the double negative there. It's worth studying, it's worth looking at, but we probably have to be more rigorous and careful and better designed in our approaches than we have been previously. Something that I think is of concern to people who are working with chemicals, because if you have an epigenetic change, what does that mean? We often think of any change from the normal situation inherently to mean something bad. But it could mean a lot of different things. It doesn't necessarily mean that there are cells that were healthy which are now becoming diseased. It could mean that you have a different mixture of cells that are present in the sample of somebody who had an exposure versus somebody who did not. And this is a sort of a technical source of variability, but it reflects an underlying cellular process that is probably a little bit too much ignored at the moment. So it may be that by studying epigenetics, epigenetic processes, we identify stuff, and that stuff is non-specific in the first instance. We see that there's a change associated with an exposure to a chemical. And as a biomarker, that's pretty okay. That may be the phenotype that you want to study, and just say, I know something's happening here, but I don't know why. But I'd like to understand what dose creates that, and whether this is passed from generation to generation and so on. Why am I seeing this change? And that's where it becomes tricky, because interpreting epigenetic results, it may mean that a certain cell type has changed its epigenome and it's now slightly different to how it was before. Or it may mean that there could be genetic differences between the individuals that you're studying, changes in gene expression, changes in the cell subtypes that you're testing between the individuals who are exposed to the toxin or not. And if you can pull out all of those effects, you will get insights into what's happening as a response to the toxin, but you'll also start to filter down on the very high confidence epigenetic changes, which then could be a real insight into the mechanism of action of the toxic exposure. So I think the future for us in this kind of work is to do the studies, do them carefully, and generate interpretable results, as opposed to just taking at face value a change in epigenetic regulation associated with the toxic exposure and saying, I assume that this is a bad thing, or I assume that this is something that is a warning sign that we should be careful about this toxic exposure. It may merely reflect a normal physiological response to the toxin exposure, because by definition you have to have a response to something that's toxic. It does not necessarily mean that the agent is dangerous. It just may mean that this is a normal response reflected by the epigenome. There's an enormously talented group of people here today, and the areas of expertise are such that you have to really dedicate yourself to toxicology to be a good toxicologist, and certainly you have to dedicate yourself to epigenetics to understand what you're doing with your molecular assays and their interpretation. And it's often very difficult to cross those silos. So for somebody like me, I listen to what the toxicologists are saying, and it's enormously useful for me, because I then understand how they're thinking, what the issues are, how they perform their experiments, what resources are available to them, and the ideas start flowing. And I can then talk to them face to face, because this is not a meeting which is all didactic. We have plenty of opportunities to interact and ask questions and discuss things with each other. And we're finding the common ground, we're bridging the silos.